Now, I would like to welcome all of you to this series of lectures whereby we are dealing with the development or embryology of the CNS or the central nervous system. Now, the first objectives, we have neurotube formation, neural crest cells origin. We have number 2 is the brain development. Number 3, spinal cord development. Number 4, meningeal development or development of the meninges. The pituitary gland development is our last objective. But this series, we are going to handle the brain development. That is the second objective. I welcome you all. Now, in brain development, we are going to learn where and when did these parts of the brain develops. We have several parts of the brain. For example, we have the pons, we have the medulla oblongata, the cerebrum, and the cerebellum, among others. Now, brain development begins, or it sets off at the beginning of the 4th week, whereby at this 4th week, the primary vesicle, the neural tube will bulge, leading to formation of 3 vesicles or 3 bulges. These 3 bulges have specific name. The first bulge, which occurs at the anterior part of the neural tube, is called the proencephalon. The second, or the middle vesicle, is called the mesencephalon, and the third vesicle is called the rhomboencephalon. Now let me show you the slide of that. Now these primary vesicles will therefore continue to bulge, thus giving rise to 5 secondary brain vesicles. We know that we began with the 3 primary brain vesicles, which will later, and that later is at the 5th week of intrauterine life, whereby these 3 bulges, these 3 primary brain vesicles, that is the proencephalon, that is the first bulge at the anterior part of the neural tube, the mesencephalon at the middle part of the neural tube, and the rhomboencephalon at the distal end of the neural tube, will therefore give rise to 5 successive secondary brain vesicles. Now, these 5 successive secondary brain vesicles have different names. They have specific names. For example, the first part was proencephalon, which was at the anterior part of the neural tube, as you are seeing in this table. The first bulge, the proencephalon, will give rise to 2 successive secondary brain vesicles, whereby the topmost part of the proencephalon will give rise to telencephalon, and immediately beneath telencephalon we have diencephalon. Mesencephalon will remain to be secondary mesencephalon. It will retain, it can't divide into 2, whereby the rhomboencephalon, which was at the distal part of the neural tube, will also bifurcate. It will also divide into 2. That is the metencephalon and the meancephalon at the bottom region. Now, these 5 successive secondary brain vesicles will give rise to different parts of the brains. For example, the telencephalon will give rise to the cerebral cortex, the cerebrum, and the white matter, while the diencephalon will give rise to the epithalamus, the thalamus, and hypothalamus. The metencephalon will give rise to brainstem, midbrain, or just the midbrain. And the metencephalon will give rise to the pons and the cerebellum. The last one at the distal part The meninges will give rise to the medulla oblongata. Hope you've heard many times being mentioned, for example, in high school, whereby we've been just talking about this medulla oblongata. Now, beneath that medulla oblongata, we know that the following is the spinal cord beneath that meninges. Now, this whole process we've been talking about is called the vesiculation process. Now, this is the simple table whereby it shows those divisions. Now, for purpose of understanding, let me just summarize in some 3 to 4 minutes there. So we've said that by week 4 of intrauterine life, the neurotube will bulge into 3 successive brain vesicles. These 3 successive brain vesicles, at the beginning of week 5, they continue bulging into 5 successive secondary brain vesicles. Now, the 3 primary brain vesicles are: the first one at the anterior part of the neurotube is called the proencephalon. The middle one is called the mesencephalon, and the distal one is called the rhomboencephalon. The proencephalon at the anterior part of the neurotube will give rise to 2 secondary brain vesicles. And remember that that giving rise of those 5 secondary brain vesicles occurs or happens at week 5 of embryonic development. Now, the first one, the proencephalon, will give rise to telencephalon and diencephalon. The middle one, called the mesencephalon, will just retain to be secondary mesencephalon. And the distal one, the rhomboencephalon, will give rise to 2, the metencephalon and the mielencephalon. And beneath it, we know we have the spinal cord, and we'll talk it shortly. Now, that is the end of the vesiculation process. So when you are told to describe the process of vesiculation process, you can just put in your raw notes. You just begin at the 4th week, the formation of the 3 brain vesicles, and at the 5th week, formation of 5 successive brain vesicles, and what each will later give rise to. Now, we now move to our second objective, that is the development of the spinal cord. The neurotube consists of 3 cellular layers from inner to outer. So the innermost layer is called the ventricular zone, or what you call the epidermal layer. The second one is the mantle layer, and the third one is the marginal layer. So from inside we have the epidermal layer, the middle one we have the mantle layer, and the marginal zone. The ventricular zone will give rise to neuroblasts, and these neuroblasts are just the future nerve cells. And it will also give rise to the glioblasts, which are the future supporting cells. These two cells will migrate. Which migrate into the intermediate zone to form 2 collection of cells. So I've said the ventricular zone will give rise to neuroblasts and glioblasts, and these neuroblasts and glioblasts will migrate into the intermediate zone, or what you call the mantle layer, in simple terms, to form two collection of cells. So after migration of this glioblast and neuroblast, it forms two collection of cells. The first one, you have the ala plate and the basal plate. And these plates, the ala plate and basal plate, are separated by what you call the sulcus limitans. Now, the cells in ala plate become afferent. What do I mean? They become the sensory neurons and form the dorsal horn of the spinal cord. Now, the cells in ala plate becomes afferent, meaning that they give rise to sensory neurons and form the dorsal horn of the spinal cord. We know that the spinal cord has the four zones, the two at the top and the two at the bottom. Now, these cells, the cells in the basal plate becomes efferent. Now, we've said that the cells in the ala plate, and the basal plate was just formed by the migration of the neuroblasts and glioblasts. Now, the cells in ala plate, we've said it give rise to sensory neurons or the afferent neurons, while the cells in the basal plate will give rise to the two obvious, the motor neurons, or what you call the efferent neurons, and they forms the anterior horn of the spinal cord. Now, the ventral horns bulge ventrally to create the ventral-median fissure, while the dorsal horn will merge to create the dorsal median septum. Let me repeat that point again. The ventral horns bulges ventrally to create the ventral median fissure, while the dorsal horn merge to create the dorsal median septum. Now, the lumen of the neurotube, and the lumen is in the central region. Now, the lumen of the neurotube becomes the central canal of the spinal cord, forming a butterfly-like structure. That's why you see when you are presenting, or when you are drawing an exact replica of the spinal cord, we show that those horns are just like a butterfly structure. Now, that is the summary of development of the spinal cord. Let me show you the slides which I have. Now, we said that the ventricular zone will give rise to neuroblasts and glioblasts, which are the future supporting cells. And these neuroblasts and glioblasts will migrate into the intermediate zone to form two collection of cells, that is the ala plate and the basal plate. And these two collection of cells, or the ala plate and the basal plate, are separated by a layer called the sulcus limitans. It's not a layer, it's a groove, separated by a groove called the sulcus limitans. Now, the ala and the basal plate cells. Now, we said that the cells in the ala plate will give rise to the sensory neurons, while the cells in the basal plate will give rise to the motor neurons. By giving rise to those successive neurons, they form the ventral or the anterior horn of the spinal cord. The two ventral horns bulge ventrally to create the ventral median fissure, while the dorsal horns merge to create the dorsal median septum. The lumen, which is at the middle of the neurotube, becomes the central canal of the spinal cord. Okay, let's proceed. Now, the spinal cord will extend through the entire length of the vertebral canal at the week 8 of the development. Now, at birth, the cornus medullaris will extend up to the vertebra, the lumbar vertebra number 3. And in adult, the cornus medullaris will extend the L1 vertebra. It's just an accessory slide. Now, we move to our third objective, whereby we are going to deal with the development of meninges. But this development of meninges is just simple and direct to the point. We know that meninges, we have Three types of meninges. We have the pia mater, the arachnoid matter, and the dura mater. And we know, the dura mater will develop on its own, while pia mater and arachnoid matter will arise from a different zone. Let's now see. Now, the dura mater arises from the paraxial mesoderm, which surrounds the neurotube. You know that mesoderm, there are two types of mesoderm. There's a paraxial mesoderm, and I hope we shall handle it. Now, the pia mater and arachnoid matter will arise, or they arises from the neural crest cells, just like that. It's simple. We say that the dura mater arises from the paraxial mesoderm surrounding the neurotube, while both the pia mater and arachnoid matter arises from neural crest cells. And that is just the development of meninges. Okay. Now, that is our final objective. In our next lecture, we are going to handle the development of the pituitary gland, or what you call the hypophysis. We know that pituitary gland, there are two types of pituitary gland. We have the anterior pituitary gland and the posterior pituitary gland. And each pituitary gland arises from different origins. So let me just continue. I think there is no need of slides. Let me just handle it in some 5 minutes to go. Now, hypophysis, or what you call the pituitary gland, arises from an evagination of oropharyngeal membrane. They arises from an evagination of oropharyngeal membrane called the Rathke's pouch, while the posterior pituitary gland arises from an evagination of neuroectoderm from diencephalon. Hope you remember the diencephalon. The diencephalon was a branch, or it arises from the first part, the anterior part of the neurotube called the proencephalon. Now we've said the anterior pituitary gland arises from an evagination of oropharyngeal membrane. This oropharyngeal membrane evaginates to form this anterior pituitary gland, while the posterior pituitary gland arises from an evagination of neuroectoderm from diencephalon. And that is the end of development of pituitary gland. It is direct to the point. Now, up to that far, I'd like to thank you for this time, and I'll just continue giving you some embryological lectures. Now, in our next lecture, to finish with this development of spinal cord, we are going to handle the clinical correlations of the spinal cord and the anomalies of the brain. That is our next lecture. It will be brief and to the point. Now, my name is Evans Kipkoech from Masinde Muliro University of Science and Technology, that is Kenya, East Africa, and I would like to continue giving some lectures based on embryology, and a little bit we can even handle the anatomy part of it. Now, hope these lectures they are of great importance, or they are of great help to medical students. That is the Bachelor of Medicine and Surgery. You have clinical medicine students. We have Bachelor of Physiotherapy. We also have the nursing students, and even and epidemiology, I think so. Now, up to that far, I want to thank you all for your cooperation, and hope you will continue following my lectures. I would like you to subscribe to my YouTube channel so that you'll receive the notifications based on my successive lectures. Thank you, thank you, thank you. very much.